Hello everyone and namaskar. This is Dr. Sudarshan Sambajara Pedge, Assistant Professor Department of Zoology, Kailaswasi Rasika Mahavidyalaya Devani, your zoology companion, and welcome to my lecture. So in the last lecture we completed the nomenclature of enzymes as well as the introductory part of enzymes. Now today we will learn about classification of enzymes. So let's get started today's lecture. So classification of enzyme I have once again repeated here this slide because we have thoroughly seen today, so that's why I have put here. So yesterday we have discussed on that topic, the classification of enzymes. In that we have discussed that is the one particular power of enzymes, that is the catalytic power, and that power it splits the molecules as well as uh, atoms as well as join with them and transfer of group in the one atom or one molecule to the another molecule. Okay, that is the one particular catalytic power of enzymes. We have also discussed in the last lecture, as well as another that is the International Union of Biochemistry has recognized 6 classes of enzymes, that is the hydrolysis, transferase, oxidoreductase, lysis, and isomerase, as well as ligase. These also discussed. So now today we will learn about thoroughly, that is the first one is the hydrolysis. First enzyme is the hydrolase. This enzyme break up the complex molecule into simpler components through the process of hydrolysis or condensation of the substrate. Okay, you have seen here in the reaction I have put here, that is the AB, one particular alphabets, and plus HOH is the react or hydrolysis form of AH plus BOH. Okay, and after the repeated this reaction, that is the condensation is occurred, that time AH again reconverted into the ABHOH. Okay, that is called as the condensation. It means the enzymes break up the complex molecule into simpler components through the process of hydrolysis or condensation of the substrate called hydrolysis. Okay, according to the classes of substrates on which they act, they have divided into 5 types. Okay, first is the proteases and peptidase, second is the carbohydrates, third is the esterase, fourth is the nuclease, and fifth one is the amidase. Okay, see one by one. First the proteases and peptidase. These are the proteolytic enzymes which brings about the hydrolysis of proteins into proteases and peptones, and then into polypeptide, dipeptide, and the amino acids called proteases or and peptidase. Okay, what is meant by the peptidase or protease? These are the one particular proteolytic enzyme and which brings about the hydrolysis of proteins into the protease and peptones, and then into polypeptides, dipeptides, and amino acids. Okay, that is called the protease enzymes. And these proteins enzymes again divide into 3 subtypes, that is the carboxypeptidase, aminopeptidase, and dipeptidase. In the carboxypeptidase, the carboxypeptidase remove the peptide linkage at the carboxyl group or carboxyl end. Okay, removal of peptide linkage. Okay, that is the one particular role of carboxypeptidase enzyme. Another second is the aminopeptidase enzyme. That aminopeptidase enzymes act on peptide linkage adjacent to the free amino acid group. Okay, that is the act on the free amino acid group, that is adjacent to the free amino acid group. Okay, peptide adjacent to the free amino acid group called with the help of aminopeptidase enzymes. Okay, and another third is the dipeptidase, and these dipeptidase enzymes act on certain dipeptides. Okay, and these are the proteases and peptidase types, subtypes. Okay, another second one is the carbohydrates. In the these carbohydrates enzyme hydrolyze the glycoside linkage of single glycosides, oligosaccharides and polysaccharides. Okay, these enzymes easily hydrolyze the glycoside linkage of simple glycosides, oligosaccharides and polysaccharide. This means it means the breakdown of glycosides, oligosaccharides and polysaccharides. Okay, so some carbohydrates, the important carbohydrates are the amylase, the sucrase, lactase and maltase. Okay, these are the subtype of enzymes in the carbohydrates. Okay, first is the amylase. In the amylase, hydrolyze starch and glycogen into maltose. Okay, amylase hydrolyze the starch and glycogen into maltose. Okay, another second one is the sucrase, and this enzyme easily changes sucrose into glucose and fructose. Okay, and third one is the lactase. That enzyme easily digests milk sugar lactose to glucose and galactose. Okay, that is the one particular function of lactase enzyme. And another fourth one is the maltase, and that maltase enzyme, it acts on maltose to form glucose. Okay, these are the carbohydrate enzymes. Okay, another third one is the esterase. In the esterase, these are the basic lipolytic enzymes which bring about the breakdown of organic esters and neutral fats to form glycerol and fatty acids. Okay, it means it is a breakdown of ester and natural fats, neutral fats to form glycerol and fatty acid. Okay, and these some known lipolytic enzymes are, there is a lipase, phosphatase and acetylcholine esterase. Okay, in the lipase is a important role in the breakdown of fats, as well as second is the phosphatase, it acts on simple and phosphoric ester. Okay, as well as number C is the acetylcholine esterase. In that acetylcholine esterase acts on the acetylcholine in the nervous tissue, forms cholines and acetic acids. Okay, this is the esterase. Okay, another fourth one is the nuclease. In the nuclease enzyme, these enzymes are concerned with the hydrolysis of nucleic acids. Okay, only hydrolysis of nucleic acid occur in the nuclease enzyme. Okay, and are of some three types, that is the polynucleotidase, nucleotidase and nucleosidase. Okay, in the polynucleotidase, hydrolysis of nucleic acid into nucleosides. Okay, hydrolysis of nucleic acid into nucleotides. This is the one particular function of polynucleotidase. Okay, another second one is the nucleotidase, and this enzyme breaks down nucleotide nucleotides into nucleosides. Okay, nucleotides into nucleotides sides. Okay, sorry, breakdown of the nucleotides into nucleosides. Okay, and another last one is the nucleoside nucleosidase. In the nucleosidase, these enzymes break down nucleosides into sugar and bases. Okay, this is the nuclease enzyme. Okay, another fourth one is the amidase. Sorry, fifth one is the amidase. These enzymes are related with digestion of amide. Okay, amidase is related with the digestion of amides. Okay, and are of three types also, that is the arginase, urease and purinamidase. Okay, arginase is a breakdown of arginine in the liver into ornithine and urea. Okay, it is a one particular important role in our liver, that is the breakdown of arginine into the ornithine and urea. Okay, that is the arginase. Okay, another second uh, urease enzyme, that urease enzyme breakdown of urea. Okay, and in last third is that is the purinamidase, that purinamide is concerned with the digestion of purines. Okay, easily digest the purine molecules. Okay, that is the hydrolase enzymes. Okay, another next is the transferase. In the transferase enzyme, the enzymes catalyze the transfer of group from one substance to another. Okay, called transferase enzyme. Okay, means the transfer of a group from one substance to another, called the transferase. Okay, transferase enzyme. So first is the transaminase. Transaminase concerned with the transfer of amino group. Okay, it means the transfer of amino groups. Okay, and another second one is the transphosphatase. This transfer phosphatase is a transfer of phosphate group, only phosphate group. Okay, and last is the transacetylase. Okay, these transacetylase easily transfer of acetyl group only. Okay, these are the transferase enzymes. Okay, so we have seen today that is the two types of uh, enzymes, classification of class two class we have studied today. It is the hydrolase and as well as transferase. Okay, uh, so remaining parts we will see later on. Thank you very much.